Let's take a minute and summarize what we've talked about so far. Glycolysis and pyruvate processing and the Krebs cycle are going to oxidize glucose to carbon dioxide. We're going to break glucose apart. We're going to catabolize glucose. And when we break the bonds, we're going to release the electrons that are in those bonds. Those electrons are going to get held onto temporarily by electron carrier molecules. And we're going to make a little bit of ATP along the way. Remember, we make ATP during glycolysis and during the Krebs cycle, but not during pyruvate processing. That ATP is being made through a process called substrate level phosphorylation. I've written out an equation here for us to summarize what we've talked about so far. It's an unbalanced equation, but just again so we think about what's going in and what's coming out. Here's our glucose, C6H12O6. We have to provide electron carrier molecules, NAD+, and FAD. We have to spend a little bit of ATP. And a whole lot of enzymes are involved. And we're going to end up with carbon dioxide, a little bit of water, and reduced electron carriers, NADH, FADH2, and some ATP. Some ATP that was made, again, through substrate level phosphorylation. In humans and other aerobic animals, the carbon dioxide that is produced from glucose is going to get disposed of as a waste gas. The question then is, what's going to happen to those reduced electron carriers? What's going to happen to the NADH and the FADH2? And that's where we will pick up now. This is a drawing of the first three sets of reactions as they occur in a eukaryotic cell. So this is a summary of glucose oxidation, glycolysis, pyruvate processing, and the Krebs cycle. And what they've drawn here is the interior of a eukaryotic cell, highly magnified. So out here, this is cytoplasm. And over here, this is a mitochondrion. So this is the outer mitochondrial membrane. This is the inner mitochondrial membrane. And you can see it's folded up inside here. That's what these blue areas represent. This area that's colored in this reddish orange color, this is the matrix. This is the space inside the inner mitochondrial membrane. Remember, there's also a space between the two membranes. That's the intermembrane space. So glycolysis is happening in the cytoplasm. Glucose is being converted to pyruvate in 10 individual steps. We're going to reduce our electron carrier, and we're going to make a little ATP. The pyruvate is then transported into the matrix, and it will be sent through pyruvate processing to make acetyl-CoA, a little bit of carbon dioxide, and we'll see our electron carriers reduced. The acetyl-CoA is then fed into the Krebs cycle. It will be oxidized to carbon dioxide. We'll reduce NAD. We'll also reduce FAD. And we'll make some ATP along the way. This is a nice summary diagram for us. It happens to be balanced, but I don't need you to learn the balanced reactions. I want you to have an understanding of what goes in, what comes out, where ATP is being made, how it's being made, and where the electrons are going, and what the electrons are ultimately used for. On this slide, we're just reminded of this process of substrate level phosphorylation. Think of substrate level phosphorylation as a piggyback kind of a reaction. The green molecule in the drawing is an enzyme, one of several different kinds of enzymes that are capable of this reaction. It's got an active site here where a substrate molecule has bound, and the substrate molecule is shown in white. Notice that there is a yellow ball at the end of the substrate molecule. That's a phosphate group. There's also another active site here where ADP is bound. As this enzyme performs its catabolic work, as it turns this substrate into a product, the phosphate group on the substrate will be transferred to the ADP and ATP will get made. So we are making ATP sort of in a piggyback reaction as the other reaction is proceeding, as substrate is becoming product. Now we are at the end of the Krebs cycle. We have finished oxidizing glucose. We have released all of the electrons from all of the bonds in that glucose molecule. And all of those electrons are being temporarily held onto by our electron carrier molecules. So we're done with the glucose. But now we need to do something with the electrons. This is what we refer to as electron transport. And remember, electron transport in humans and other animals is taking place in the mitochondria, in the electron transport chain. So the reduced molecules NADH and FADH2 are going to become oxidized again. They're going to lose those electrons that they're carrying. They're going to release them into the electron transport chain. The electron transport chain is actually a series of proteins, and they're very particular, very special proteins that are grouped together into these large complexes. And each of them has a different electronegativity. The electron transport chain is going to receive the electrons. It's going to gain the electrons from the electron carriers. So it's going to get reduced at the top of it, if you will. And then it's going to pass the electrons from one protein to another, to another, to another, down the electron transport chain. I like to imagine the electron transport chain as a staircase. The electrons come in at the top of the staircase, and then they're passed down, step by step by step by step. It's a series of oxidation reduction reactions here, as the electrons are passed down the electron transport chain. At the bottom of the electron transport chain is the final electron acceptor molecule. And again, for aerobes, like humans, that's going to be oxygen. So the last protein complex in the chain will gain the electrons, it will get reduced, and then it will be oxidized as it hands them off to oxygen, and the oxygen will get reduced. And the reduced oxygen, along with some hydrogen, will become water. Now, why do we bother passing electrons down through this electron transport chain? Why not just have the electron carriers hand the electrons directly to oxygen? Well, the answer is that the electron transport chain is capable of harvesting the energy out of these electrons as they pass down the chain. So this energy is critical, because the energy is what we will need to make ATP ultimately. The energy will be used to create the hydrogen ion gradient that will then be used to build ATP. So the energy that is harvested from the electrons in the electron transport chain will be used to pump protons across the inner mitochondrial membrane into the intermembrane space. So remember where we are in the mitochondria. We are inside the matrix. We're in the inner chamber of the mitochondria. We're going to pump protons, hydrogen ions, from the matrix across the inner mitochondrial membrane into the intermembrane space. Whew, lots of terms there. This is going to create an ion gradient, and ion gradients have potential energy. That ion gradient is then going to be allowed to resolve. It's going to be allowed to go away as the hydrogen ions flow back across the membrane into the matrix. And they're going to flow through that molecule called ATP synthase. It's an enzyme. And as the hydrogen ions flow through the ATP synthase, they're going to cause it to literally spin, to rotate, to turn. And the kinetic energy of that rotating motion is going to create ATP. On this slide, we're looking at a little piece of membrane that is representing the inner mitochondrial membrane. So we're inside a mitochondrion here. The outer membrane would be out here, out of our vision. Here's the inner membrane. So here's the intermembrane space. And over here, this is the matrix. So we brought pyruvate into the matrix, and it went through pyruvate processing, and then it went through the Krebs cycle. And the electron transport chain, you can see, is embedded right in the inner mitochondrial membrane. So let's take a look at what's happening. NADH and FADH2 are coming in and donating their electrons to this complex chain of proteins. Now you'll notice there's a 1 here and a 2 and a 3 and a 4. There are 4 complexes of protein. So number 1 complex is actually a group of proteins. Number 2 is a group of proteins. You can also see that NADH actually donates to complex 1, FADH2 donates to complex 2. Don't worry about those details. Just know that these electron carrier molecules are going to donate their electrons. They're going to lose their electrons into the electron transport chain. They are getting oxidized. The electron transport chain gets reduced. And then the electrons are passed. They're passed through this complex. And along the way, their energy gets harvested. The energy is going to be used to pump hydrogen ions out of the matrix and into the intermembrane space. The electrons themselves at the end of the chain will be received by oxygen. So oxygen is going to get reduced, and we're going to make some water. The electron transport chain is embedded in the inner mitochondrial membrane, and it's made up of a whole bunch of different proteins that are grouped into these 4 complexes, numbered complexes 1 to 4. These proteins are unusual proteins. They contain very specific, very unusual chemical groups that affect their electronegativity. They have to be good at passing electrons. So they contain things like flavins and iron-sulfur complexes, heme groups. Don't worry about these details so much. Just understand that the proteins in the electron transport chain have varying electronegativities, so they're able to pass electrons one to another. There is another molecule we need to talk about, another molecule that is in the inner mitochondrial membrane, but this one is not embedded in the membrane so much as it moves around inside the tail region of that membrane. This is a very interesting molecule called ubiquinone. Ubiquinone. So ubiquinone is not a protein. It's a type of molecule called a quinone. And because ubiquinone is found in many, many, many different organisms, many different eukaryotic types of cells, the people who discovered it decided to name it a combination of its being ubiquitous or common and being a quinone. So that's where it got the name ubiquinone. Now, just to make things a little more confusing for us, ubiquinone also goes by the name coenzyme Q. And some references just call it Q. So all three of those mean the exact same thing. Ubiquinone, coenzyme Q, or just plain Q. The next time you go into a drugstore, go over to where the vitamin mineral supplements are, and you'll find coenzyme Q sold as a nutritional supplement. Now, what makes Q interesting is that it is lipid soluble. So it literally sort of lives in the tail region of the inner mitochondrial membrane, and it can move freely within those tails. Q also acts as a pump, so stay tuned on that one just a minute. In contrast to Q, the proteins that make up the electron transport chain are embedded. They stay put, and that's because we need those protein complexes to stay in order so that they can do their job correctly. Each of the protein complexes, as it accepts electrons, it then needs to pass those electrons on to the next protein. And because those proteins are embedded, it can be a little bit difficult to manage that. That's where Q comes in. Q actually takes the electrons from one protein complex and passes them over to the next protein complex in the electron transport chain. Take a look at this drawing of the electron transport chain. So this membrane here represents the inner mitochondrial membrane. The outer mitochondrial membrane would be out here somewhere in this drawing. Here's the intermembrane space in blue, and here is the matrix in that that orangish brown color. The electron transport chain is drawn as these green complexes, numbered 1 to 4. So NADH and FADH2 come in and donate electrons to the electron transport chain. And those electrons are going to get passed from complex 1 to complex 2 to complex 3 to complex 4 and finally to oxygen. Now, along the way, the electron energy is being harvested. And that energy is going to be used to build a proton gradient, and they're showing us that gradient building up in the intermembrane space. Here is our friend Q, shown in orange. Q is going to pick up electrons from one complex and carry them over and donate them to the next complex. Now, here's the other work that Q can do. Q can act as a pump. So Q can actually help carry protons across the membrane as well. You can see some protons are traveling across the protein complexes. They're being pumped with the protein complexes, but they're also being pumped with Q. Now there's one more molecule we need to know about, and that's cytochrome C. Cytochrome C is a little bit like Q in that cytochrome C is going to help to transfer electrons between the protein complexes in the electron transport chain. If we go back to the picture we just looked at, you can see cytochrome C right here. Now, cytochrome C technically does the work of transferring electrons from complex 3 to complex 4. Don't worry about that detail. Just remember that both Q and cytochrome C are able to assist in transferring electrons down the electron transport chain. As those electrons are transported, their energy is being harvested, and it's pumping protons, hydrogen ions, into the intermembrane space, creating an ion gradient. There's energy in that ion gradient, potential energy in that gradient. And that energy is going to be used to make ATP. The gradient is allowed to resolve by having the protons flow back into the matrix through the molecule ATP synthase, which is also embedded in the inner mitochondrial membrane. The energy from that ion gradient, as the gradient is being resolved, is going to be transferred to the ATP synthase. It's going to make the ATP synthase spin. And that process, that spinning motion, is what's necessary to build the ATP. Making ATP this way, and by that I mean using an ion gradient, the energy in an ion gradient, to build ATP using ATP synthase. That is what we call oxidative phosphorylation. This process, oxidative phosphorylation, is the most efficient way to make ATP in a living cell. From that one glucose that entered in, the cell is going to get between 30 and 36 ATP molecules produced. That is a tremendous return on an investment. So oxidative phosphorylation is very efficient. Compare that to substrate level phosphorylation, which is inefficient. Remember, that glucose that was entered into cellular respiration, we got a couple of ATP made in glycolysis through substrate level phosphorylation, and we got a couple of ATP made during the Krebs cycle through substrate level phosphorylation. But we're going to get 30 to 36 ATP from oxidative phosphorylation. It's a great way to make ATP. This will end part 1 of our discussion of cellular respiration. In the next video lecture series, we'll discuss fermentation.